Today we will learn the chapter 5. Chapter 5 is describe the extension and modification of basic principle. Basic genetic principle. Basic genetic principle, the extension and modification. First one is we will learn the additional factors at a single locus can affect the result of genetic crosses. And second one, we will learn about genetic interaction, and also we will learn about sex influences the inheritance and expression of gene. Finally, we also learn some environmental effect which can affect the expression of the genotype. In this slide, if you see the direction of shell coiling in the snails, snail is 달팽이. If you see the direction of shell coiling in the snail, one is the left-handed, the other one is a right-handed. So a little difference between each other. So maybe we will see some detailed information in direction of shell coiling at the end of this chapter, but direction of shell coiling in snail is determined by genetic maternal effect. So, okay, we will see the details in the later, okay? In this slide, we will learn the additional factor at a single locus can affect the result of genetic crosses. So, okay, the genes at the same locus, there are two versions of the same gene. For example, the large R or small r. Example is a large R or small r. So each version of the same gene defined, we call it as allele, large R allele and small r allele. So we already learned the concept in chapter 3, the allele and the genes. But at single locus, previously we only learned that at single locus there is no additional factor that affect at single locus, but we will see another additional factors at a single locus, which is the type of dominance. Okay, in the type of dominance, there are three subtypes. One is complete dominance. We already learned in previous chapter 3. Now we will learn the incomplete dominance, and also we will learn the codominance. So what is complete dominance? So this one, so for example, this flower is the A1 equals red color. This homozygous allele equals red color, and also this additional A2 homozygous is equals the white color. So after crosses, they will make the A1 and A2 or A1 and A2. So this is heterozygote. So if the heterozygote is red color, it means that A1 allele is dominant than A2 allele. So if the this heterozygote is white, the A2 allele is dominant than A1 allele. So this is we already learned that complete dominance example. Okay, so now we will see that in some incomplete dominance. So in F1 generation, they will make the A1 and A2 heterozygote. So if the phenotype of the heterozygote is a forms between the phenotype of the two homozygous. So one homozygous is red color, one homozygous is the white color. So the phenotype is falls between the red and the white. So in these cases, we call it as dominance is incomplete. So this is the example of incomplete dominance. So Based on these findings, we can find that additional factors at a single locus. What was additional factor? One is complete dominance or incomplete dominance. Now we will see the later the codominance is later. So it can affect the result of the genetic crosses. If the complete dominance, they will only show that red color and white color. But they are when they are the incomplete dominance, you will see that the phenotype will be the difference. Okay, the result of genetic crosses will be different. Okay. Now, so in this slide, we will learn the incomplete dominance. Okay. So phenotype of the heterozygote is intermediate, means that falls within the range between the phenotype of the two homozygotes. So one example is the fruit color in eggplant. What is eggplant? In Korean, we call it as gaji. Gaji. Okay. In in the parent generation, the one parent showed that purple fruit means that the large P and large P. Another one is a white fruit, and the genotype will be small p and small p. So they will make the gamete 배우자 like the light p or small p. So after fertilization, the F1 generation, they will have the large p, small p. But the phenotype is not purple or is not white. The phenotype of heterozygous is intermediate, means that falls within the range between the phenotype of the two homozygous, so violet. So after that, they did self-fertilization. So this is the F2 generation. So this is a Ponnett. So one will be large P and small p, and one will be large P or small p. In Ponnett scales, in this scale will be the large P means that the phenotype will be the purple. How about this one? So this one will be the large P and small p, the phenotype will be the violet. How about this one? It's also large P, small p, the phenotype will be the violet. How about this one? This one is a small p, the phenotype will be the white. So the conclusion was the genotypic ratio is 1 to 1, and also phenotype ratio will be the 1 to 1. Based on this result, fruit color in eggplant is inherited as an incomplete dominant trait. So if the color of the fruit color in eggplant is inherited in dominant trait, complete dominant trait, maybe the phenotype will be 3 to 1. 3 will be the purple and 1 will be the white, but the fruit color in eggplant is inherited as an incompletely dominant trait. Their phenotype ratio is 1 to 1. In this slide, we will learn codominance. Previously, we learned incomplete dominance, but this is codominance. Codominance means that the phenotype of heterozygote include the phenotype of both homozygotes. Example is MN blood type. So we are very familiar with the ABO blood type, but there is also MN blood type is also available. For example, the genotype is the LM is the blood type is the M. How about the N? LN is the blood type is N, but when the heterozygote LM, LN existed together, the phenotype of heterozygote shows that both phenotype of the both homozygous. So it can show the M and blood type. So, for example, blood type M means that antigen present on the red blood cell is M. How about N? N means that the antigen present on the red blood cell is N. How about the LM? LM means that the antigen present on the red blood cell is M and N. So this is the red blood cell. You see here M blood type show they have the M antigen. How about N? They have N antigen. MN means that they have both antigen. That's why this is the experiment to show that reaction with antiserum, for example, this serum has the antibody against the M antigen. So in this case, this serum can react with red blood cell of this one, so they can be aggregate. They aggregate like this. How about the anti-N serum? This is an antibody against N antigen, but this blood type, they don't have any N antigen, so there is no reaction, no reactions like this. How about this one? So anti-M serum cannot react with the N blood type, but anti-N serum can react with the N antigen, right? So they can be aggregated. They aggregated. How about LMN? So MN phenotype. So MN blood type. So this one is this because they have the co-dominance. They have both type of the antigen, so they can be aggregated. When they react with the anti-M antibody, so they can be there is a reaction here, and also when they add anti-N antibody, there is also another reaction like this. Okay, so this is we call it as co-dominance. Once again, the heterotype of the heterozygote phenotype includes phenotype of both homozygous M and N. Okay. So this is the table 5.1 to summarize the difference between the complete dominance and incomplete dominance and codominance. So what is complete dominance? So complete dominance means that phenotype of heterozygotes is the same as the phenotype of one of the homozygous. How about incomplete dominance? Phenotype of the heterozygote is intermediate between the phenotype of the two homozygous. How about codominance? Phenotype of the heterozygote includes the phenotype of both homozygous. This is we call it as codominance. So in this slide, we will learn additional factor. We call it as a level of phenotype may affect the dominance. The very famous example we can see in the CFTR gene. CFTR gene full name is cystic fibrosis transmembrane conductance regulator. CFTR is a channel in the membrane that regulate chloride ion. So usually in the lung cell inside, they have chloride ion, and after that, usually in the chloride ion, the chloride CFTR is moves out the chloride ion. So usually in these cases, in the lung cell, there are lots of the cilia like this. So when chloride ion is moves out, in these cases, cilia can move very well. But when in the cystic fibrosis patient, CF patients, there is mutation in the CFTRs. So there is no conductance of chloride ion outside. In these cases, in CF patient, mutated channel stays closed, so chloride ion is built up in the cell. They are built up in the cell, not can go outside. Okay, so in these cases, they have thick and sticky mucus, and the cilia cannot move. Okay, how about in the healthy? Usually in healthy, this is lung. So this is the CFTR channel. So usually the chloride ion can go outside, and also this channel usually inhibit the activity of the sodium channel. So sodium can also stays outside of the cell. So in these cases, the water can go outside and go inside very well. So usually the mucus is located here, but they have some space here. So that's why cilia can move very well. But in these cases, when CFTR does not working, usually the chloride ion is built up inside the cell, and also sodium ion is working very well. So inside the cell, there are lots of sodium and chloride ion. In these cases, the water can go in, so there is no water. So in these cases, and also they have thick and the sticky mucus, so cilia cannot move. They can be stuck. But in the heterozygote, the allele for normal F, large F, and the defective CFTR, small f, both of them are codominant. They are codominant. But in heterozygote, functional CFTR is made to prevent cystic fibrosis. So when this is wild type, normal, and this is defective, in heterozygote, both of them is existed together, but they are codominant. But in these cases, the normal CFTR is half the mutated CFTR, so chloride can move to this region, and sodium can move to this region, and in this cases, the water can be stayed in this region, so cilia can move here. So in these cases, the level of phenotype. What is the level of phenotype? Normal phenotype is superior to the level of defective CFTR, That's why in heterozygote, they are normal. But in genetically, they are codominant. But level of phenotype is affect the dominance. So in these cases, they are heterozygote. In these cases, they are normal. For example, small f and small ff, they will have the cystic fibrosis. Okay. Okay, so now in this slide we will see also another additional factor that affect the result of genetic causes. One is penetrance and one is expressivity. Okay, what is penetrance? Okay, for example, large B is the dominant than small b. For example, large B, they show the blue phenotype like this, large BB, and small bb, they will have the some, or they will have the white phenotype. So when they cross to each other, they will make the large B, small b. Usually, theoretically, all of offspring should have the blue color. So this is the complete penetrance. But some cases they have some incomplete penetrance. For example, the total is 9, the offspring is 9, but some of them they express the blue phenotype, and some of them they express blue phenotype, and some of them they express blue phenotype. So in these cases, we call it as incomplete penetrance. So some individuals do not express the phenotype even though they inherit the alleles. So in this case, is also inherited the allele, but they did not express phenotype. This one is also, and this one is also. So in this case, we call it as incomplete penetrance. So penetrance means that the percentage of individuals having a particular genotype that express the expected phenotype. Okay. So in these cases, what's the penetrance? 100%, isn't it? How about in these cases? Only 5 is expressed the expected phenotype. It means that the penetrance will be the 50%. Okay. What is, okay, next we'll go to the expressivity. So expressivity means that the degrees to which a trait is expressed. So very famous example is a human polydactyly 다지점. Okay, so means that usually we have five finger like this, but uh, people which has the polydactylity, so it means that some people they have two external digit, or some people they have one external digit, or some people they have three external digit. So it means that human polydactyly is a human 다지점 is an example of variable expressivity. So some people have a one, some people has the two, some people has a three external digit. Okay, so this is the we call it as expressivity. Okay, this slide we will solve concept check 1. The question is that assume that long fingers are inherited as a recessive trait with 8% penetrance. Two people heterozygous for long fingers mate. What is the probability that their first child will have long finger? Okay, how about this one? So the two people, and one is a large L and small l, and the other people are also heterozygous, large L, small l. So after they mate, this is Punnett scale. One is a large L, small l, and the other is also large L, small l, and this scale is large LL, and this scale is large L, small l, and this scale is large L, small l. this is small ll. So in this case is large ll will have short finger, and the large L and small l will show the short finger, and large L, small l also will show the short finger, and small ll will have the long finger. So what is probability that their first child will have a long finger? So if the penetrance is 100%, the probability will be 1 out of 4. But because they have the 80% penetrance, so we have to multiply 0.8 and multiply 100, so it will be 20%. So what is the probability? It will be the 20% that their first child will have the long finger. In this slide, we will see another additional factor that affect the result of genetic crosses. Another genetic factor is the lethal alleles. So lethal allele cause a death at an early stage of development. So some genotype may not appear among the progeny, among the baby. So lethal allele can affect the Mendelian genotypic and phenotypic ratio in the progeny, in the baby. Okay, in the parent generation, the mouse has the yellow color. So, and they are heterozygous, large Y and small y. And another mouse is also yellow, and their genotype is large Y, small y. So after meiosis, they will make the large Y, small y, and also they will make the large Y and small y. So after fertilization, so we will have the F1 generation, large YY, and the large Y, small y, and small y, small y. The ratio, the genotypic ratio is the 1, so 1 to 1, isn't it? So you can do the Punnett scale, okay? But in the large YY cases, they are lethal allele. So if they have the large YY together, in this case is the embryo. This lethal allele, which has the large YY together, can be a lethal allele and cause death at an early stage of development. So so we only can see the ratio 2 to 1. So conclusion was the large YY mice dies, and so 2 out of 3 of progeny are yellow, and 1 out of 3 progeny are non-yellow. So this one will be the genotype will be the large Y and small y, because the large YY is the lethal allele, so they cannot survive. So based on the lethal allele, the 2 to 1 ratio is produced by a cross between 2 yellow mice. So the 2-2 ratio is a result from a lethal allele. So if your progeny have the ratio is a 2 to 1, in this case is this result is maybe result from a lethal allele. In this slide, we will solve the concept check 2. The question is the cross between 2 green corn plants. 2 green corn yields 2 out of 3 progeny that are green and 1 out of progeny that are white. The question is that what is the genotype of the green progeny and the white progeny? So now we found that ratio is the 2 to 1. 2 1. In these cases, we have to think about the lethal allele, because the lethal allele 2 to 1 ratio is usually the result from a lethal allele. So what is genotype of the green progeny? Maybe green progeny will be the large G and small g, and white progeny will be the small gg and maybe large gg, and this is will die because the G is lethal allele, so the homozygous of the large allele, large G will induce the death at early stage of the embryo. So if the ratio is 2 to 1, 2 to 1, you have to think about lethal allele. Okay? In this slide, we will see another additional factor that can affect the result of genetic crosses. Another one is multiple alleles. Usually the four genes at the same locus, usually the two alleles is present, but in these cases, more than two alleles are present. The very famous example is the dog feeder pattern. So usually there are three alleles determine the type of the feeder. So one is the MR, large MR. So R, large R means that the restricted patterns, they looks like the brown. The feeder looks like brown. And but large M, R is the dominant than the large M. Large M is mallard. In Korean, we call it as a 청동오리 pattern. They have the brown in the front, and they also have the green color in the head, and also they have the gray color at the tail. So this is the M. So M allele is the mallard. In Korean, we call it as a 청동오리 pattern. And also M is the dominant than small md. D means that dusky. Dusky means that in Korean we call it as the 거무스름한 pattern. There is no nearly similar to mallard, but they don't have the brown in the upper in the front side. They don't have the brown color. So this is dusky. But once again, M large R is dominant than large M, and M is dominant than small md. Okay, in this cases, when the genotype is the MR, in this cases the phenotype will be the restricted. How about in these cases? The large MR and M, in this case is also phenotype will be the restricted, 제한된 pattern. How about the MR and small md? In this case is also restricted because the large MR is dominant than large M, also dominant than small md. How about large MM? This is the mallard, 청동오리 pattern, and also large M, small md is the mallard, it's also the 청동오리 pattern. How about small md? Small md, this is dusky. 거무스름한 pattern will shows like this. So, okay, in this case is the the parents, the father was large MR, small MD in these cases. So they will show that restricted pattern means that their feeder is brown, yellow brown. How about the the father? Father is the mallard, and the genotype is large M, small md. So in these cases, their gamete in sperm, one will be the large M, one will be the small md. So in these cases, the female, they will have the egg, one egg have large MR, and one egg will have small md. So after the fertilizations, so we can do Punnett care. It's okay. In this case, in first case will be the large MR with large M. Another case is large MR is small m small d. Another case is that uh, small md and m, small md and m, so like this. Another case is the small md and small md, small md and small md. The first case is large MRM. In this case, is the color will be the restricted 제한된 pattern. How about in these cases? Comes from the large MR and small MD. This case is also restricted. How about these cases? Large M, small MD. This will show that mallard 청둥오리 pattern. How about the final cases? Small md and small md. In these cases, the color will be the dusky. They don't have a brown plant. So when the multiple allele is involved in the genetic process, the progeny will have the two out of four is restricted, 1 out of 4 mallard, and 1 out of 4 will dusky. So once again, father is also heterozygote, and mother was also heterozygote. So in this case, if only 2 allele is present, and if in the case only 2 allele is present, so after heterozygous fertilized together, after that usually the pattern will be 3-to-1. But this is in this case is multiple allele is involved, so the ratio is the 2-1-1. Okay. So in this case is when the ratio is 2-1-1, maybe it means that multiple allele is involved in the genetic process. Okay. This slide will also learn another cases in multiple alleles. The example is ABO blood type. There are 3 alleles involved in ABO blood type. One is large IA. Large IA is encores A antigen, and the large IB is encores B antigen, and also another allele is small I. Small I is encores no antigen. So large IA is dominant than the small I, and the large IB is also dominant than small I. But between the large IA and the large IB, they are similar to each other. There is no dominant and there is no recessive. In this case, so when the phenotype is A, means that the genotype will be the large IAA, large I, large IA, and large IA, or large IA, small I. Maybe so. This is genotype is like this, and the antigen type will be the large A. How about the blood type B? The genotype will be the large IB, large IB, or the genotype will be large IB or large IB and small I. And the antigen type will be the B. How about O? O will be small I and small I. The antigen type will be none. How about AB? Because there is no dominant and there is no recessive between the IA and IB. So blood type AB means that large IA and large IB, and the antigen type is A and B. So ABO blood type is also another example of the multiple allele, 3 alleles, large IA and large IB and the small I. So in this slide, we will learn another subject is gene interaction. So gene interaction is take place when a gene at multiple loci determines a single phenotype. So gene interaction means that effect of gene at one loci is dependent on the presence of the gene at the other loci. Or it will be difficult to understand. This looks like the dihybrid crosses. For example, dihybrid crosses means that examine 2 traits at a time. So at that time, we learned principle of independent assortment. For example, they examine 1 trait which encodes the shape, and also they also learn examine another the genes Y, so which encodes the color. So they examine 2 traits at a time. So at that time we learned the principle of independent assortment. But gene interaction means that effect of gene at one locus is dependent on presence of gene at the other loci. So it means that, for example, gene interaction means that 2 genes, for example, if there are 2 genes, more large R, large Y, this kind of 2 genes determines a single phenotype. This is difference between the dihybrid crosses. In this case, each gene encodes a trait, each gene encodes trait, but in this case, each gene determine and each gene is involved in a single phenotype. For example, in this case is fruit color. Fruit color in the pepper, okay? So, and Y is the encodes one enzyme. For example, the first step in the pathway of fruit color making. And C is located in another chromosome. So Y is for example, Y is located in chromosome 1, and C is located in the chromosome 2, something like that. But C is encodes a different enzyme, the last step in the pathway of fruit color making. So when the genotype is Y plus and C plus, color is the red color, and Y plus CC means that peach, and YY, small YY, and C plus is orange. and small yy and cc will be the cream. Color is cream. So when we learn the principle of independent assessment, the gene encodes the shape is located one chromosome and the gene encodes color of the P is located in another chromosome. So they did the independently assorted. At that case is the final the phenotype was 92321, 9232321, and in this case is also okay in the parent generation is homozygous y plus y plus c plus c plus like here is a large R, large Y, and the another parent is small y, c, c. After that, they did the F1 generation. The F1 generation is a large Y plus small y and C plus small c. So in these cases, color is red color. Okay. So at that they will do the self-fertilization. So because the Y and C is located on each is different chromosome, so they will do the independent assorted. At that, their phenotype will have the 9 will be the red color, and 3 will be the peach color, and 3 will be the orange color, and 1 will be the cream color. So this is the gene interaction. Gene interaction take place in these cases when genes at two loci determines a single phenotype. What was single phenotype? It's fruit color in the pepper. Okay. The difference between the dihybrid is in dihybrid cases, we found that one gene is encodes one trait and another gene is encodes another trait, but in this case, it's two genes encodes a single phenotype. In this slide, we will learn another gene interaction. We call it as epistasis. Epistasis means that one gene masks the effect of another gene. So one gene A, for example, A라는 진이 B라는 진이 이제 활동을 나타내는 것을 완전히 없애버리기도 하고 없애버리기도 한다 이 말이에요. One gene masks the effect of another gene. So there is the recessive epistasis and also there is dominant epistasis and also there is duplicate double epistasis and duplicate recessive epistasis. Okay, let's start from the recessive epistasis. So it means that recessive epistasis means that presence of two recessive allele inhibits the expression of an allele at a different loci, different locus, different location. Once again, presence of two recessive allele inhibits the expression of an allele at a different locus. For example, coat color in this dog, usually the dominant allele large B encodes black color, and the recessive allele small b encodes brown color. Recessive allele B encodes the brown color. But allele at the second locus, another allele is E and another allele is E. Allele at second locus affect the deposition of the pigment in the hair. For example, if there is a large E, so large E allele allows dark pigment to be deposited. black or brown. If there is a large E, it allows dark pigment to be deposited. But if there is a recessive allele small ee, it prevents the deposition of dark pigment, causing the hair to be yellow like this. Okay. Now we are examining the recessive epistasis. Presence of two recessive allele inhibits the expression of an allele at different loci. So presence of the recessive EE, it inhibits the expression of the large B or small b. So because the large B encodes the black pigments, but if there is a small EE, the color will be the yellow. And small BB is encodes the brown pigment, but so small BB, because there is a small EE, the color is the yellow. Okay, so B is dominant than the small b. Okay, if there is the B, large B, so and if there is a large E, the color is black. And also if there is a small BB and if there is a large E, color is brown, it's like this. Okay. So now is here. So parent generation is the large BB, large EE, and another parent was small BB, small EE. The one phenotype is the black, another phenotype is because there is small EE, another phenotype is yellow. After crosses, F1 generation is a large B, small b, large E, small e. In this case, what is color? Color is black. F1 generation is black. After that, they will do the intercrosses. After that, they will have the F2 generation. The ratio will be 9 to 3 to 3 to 1, 9 to, and this one can go to the here, 9 to 3 to 3, or this one can go to the here, 3 to 1, but in this case is large B, large E will encode the black color. How about small bb, large E encodes the brown color. But in this case is large B, small ee, it will encode yellow color. How about here? Small bb, small ee will encode yellow color. So the phenotype ratio will be the 9 to 3 to 4. So if they are associated independently, like here, they are associated independently, their phenotype will be the 9 to 3 to 3, 1, but there is a recessive epistasis. So in this case, it's 9 to 3 to 4. Okay. This line, we will learn another recessive epistasis. The example is expression of ABO antigen is dependence on the allele at the H locus. So they are in the dihybrid crosses. The gene encodes ABO antigen is located, for example, chromosome 1. Maybe the gene encodes the H locus. The gene allele H is located in the chromosome 2. So they will follow independent assessment. They will follow the independent assessment. Maybe their final phenotype will be the 9 to 3 to 3 to 1, but the dominant H is encodes an enzyme that converts an intermediate to compound H. So this is the intermediate. So if there is the dominant H, so they will add another sugar. So this becomes the compound H. So after that alleles at the ABO locus determines which type of terminal sugar are added to the compound H. For example, if they are gene enclosed IA, so it means that their terminal sugar will have the A antigen. How about if they are IB? In this case is their terminal sugar will have the B antigen. The color is different. So if they have the small II, in this case is no antigen can be added here. Okay, how about recessive allele? So recessive allele HH failed to convert intermediate into H, so there still be the intermediate. In these cases, there will be no A antigen and there will be no B antigen. So in these cases, even though the genotype is large IA, in this cases, if they have the small HH, all of the ABO phenotype will be the O. So, for example, large B and small I, in this cases, usually the ABO phenotype will be the B, but because they have the small HH, so all of ABO phenotype is O. Okay, for example, once again, if they have a large H, in this case, if they have a large IA, in this cases, ABO phenotype is A. How about this one? Large H and also they have a large IA and small I, their ABO phenotype is B. They will do something like this, but if there is the recessive allele small HH, it failed to convert intermediate to the H compound H, so the terminal sugar cannot be added into at the end. So it means that in these cases, the anti-ABO phenotype will be the O, even though they have the IA and IA, the genotype. Their ABO phenotype will be the O. This slide, we will learn another gene interaction. We call it as dominant epistasis. So in this case, is only a single copy of allele is required to inhibit the expression of allele at different loci. So this allele, for example, this allele is located in chromosome 1, and another allele is located in chromosome 2. So they are located in the different chromosome, so they will follow the principle of independent assortment. For example, so this is very difficult to understand. The one example you can see in blue color in summer squash. Squash is the OE. Okay, so they have the white color, and they also have the green color, and they also finally they also have the yellow color. So usually in the fruit color making, so usually the enzyme 1 and the enzyme 2 is involved in the color making. For example, a single copy of allele, dominant allele large W allele is dominant epistatic to large Y and small y. So it means that single copy of the dominant allele W is sufficient to inhibit enzyme 1 activity. Okay, if enzyme 1 activity, if the enzyme 1 activity is inhibited, the compound A cannot be changed to compound B. It means that compound A cannot be changed to compound C. So this is the dominant allele W. They are dominant epistatics to large Y and small y. How about large Y allele? Large Y allele is the dominant than small Y allele. The function of large Y allele encodes enzyme 2, which convert compound B to compound C. Compound B is the green color and compound C is yellow color. But Y is dominant than small y. So, okay, if the plant with genotype Y produces enzyme 2, which convert compound B into the compound C. For example, plants with genotype small yy do not encode the functional form of enzyme 2, so compound B cannot be changed to compound C. It means that their color will be the green. Okay, so when plants with the genotype is the small w produces enzyme 1, which convert compound A is colorless into compound B is green. But dominant allele large W is dominant epistatic to large Y and small y, so they cannot inhibit conversion of A into B or A into C. So, for example, the conclusion was the genotype large W and large Y, if they have large W, or if they have large W, small yy, and do not produce enzyme 1. So do not produce enzyme 1, the color will be the white. And also small ww and small yy, in these cases the produces enzyme 1 but cannot produce the enzyme 2, so the color will be the compound B, means that the green. How about this one? Small yy and large Y can produce both enzyme 1 and enzyme 2, and color will be green. In this slide, I will calculate the dominant epistasis. As I mentioned earlier, the dominant epistasis is a dihybrid crosses. Each gene is located in different chromosomes. Usually they follow the principle of independent assessment. So parent generation was the plant with the white squashy, and also they have these parents have the large W, and large Y, and large Y. Another parent is plant with the green squashy, small w, and small yy. So this is the green. So after crosses, upon generation is the large W, small w, large Y, and small y. What is the phenotype? This phenotype, if they have the large W, the phenotype is the squashy, white squashy. So phenotype is white. After that, they did intercrosses. So because they will follow the principle of independent assessment, the ratio will be this one will be the 9, this one will be the 3, this one will be 3 and 1, 9, 3, 3, 1. But how about when the W, large W, large Y, and also large W, small YY is the white Scotchy. So the phenotype, this one will be the 9 to 3, means that the 12, 9 plus 3, 12. How about this one? This one, small WWY, means that this can make the yellow Scotchy. So yellow Scotchy. How about this one? Small WW, small YY. This one will make the green Scotchy. So the ratio will be 12, 3, 12, 2, 3, 2, 1. So usually in dihybrid crosses, if they follow the principle of independent assessment, the two trait is the assorted 9 to 3 to 3 to 1, but in this case they are also follow dihybrid crosses, but they also follow the dominant epistasis rule, so the ratio will be 12, 3 to 1. We will learn another gene interaction. We call it as the duplicate recessive epistasis. It means that two recessive alleles at either of two loci are capable of expressing a phenotype. What's the difference between the duplicate recessive epistasis between the duplicated recessive epistasis? So we can go back to the recessive epistasis. Okay. So presence of the two recessive alleles inhibits the expression of an allele at different loci. This is the recessive allele. So for example, in A locus, if they have the recessive allele, it means that if they have the two recessive alleles in one locus, it can inhibit the expression of the allele at different locus. This is recessive epistasis. How about this one? This one is the two recessive allele chromosome 1 and chromosome 2, either of two loci, this one or this one. So this is the duplicate recessive epistasis, capable of expressing a phenotype. The very famous example is albinism in snail. So for example, the small allele which has the small a is located, for example, chromosome 1, and also B is located in chromosome 2. This is the, maybe they will follow the principle of independent assortment. But when they have the recessive allele small aa, in these cases, they fail to make enzyme 1. So if they have another small recessive allele, small bb, it also failed to make the enzyme 2. Okay, let's go to here. So this is the snail. Because in this snail, they don't have any color. And this snail also, they don't have any color. This one is they have a color, brown color. So for example, the dominant allele at the A locus is required to produce enzyme 1, but which convert compound A to compound B. But compound A, they don't have any color. And compound B also, they don't have any color. So after B was made, the dominant allele at B is required to produce enzyme 2, which convert compound B into compound C, which has the pigment like brown color like this. So pigment snail must produce enzyme 1 and enzyme 2, which requires A slush A B slush. But here, so albinism arises from the absence of the enzyme 1. Enzyme 1 means that if their genotype is small AA and large B, large BB or large B small b, in these cases, so compounds B is never made, so they will show the albinism, they will show the white color. How about this one? Albinism arises from the absence of the enzyme 2. Enzyme 2 means that the small bb, so large A, small bb, or large A, small bb. So the compound C is never produced, or albinism is arises from the absence of both enzyme 1 and enzyme 2. So in these cases, we call it as duplicate recessive epistasis. So two recessive small aa or small bb at either two loci is capable of expressing a phenotype. Small aa or small bb or small aa, small bb, they can capable of express a phenotype. So in this case, we can also do calculation. For example, the large A allele and B allele is located in the different chromosome, so they will follow the principle of independent assessment. The ratio will be 9 to 3 to 3, 1. For example, in this case, one parent has the small AA, large BB. So phenotype is albino because they have small AA. Another parent is the large AA and small BB. This is also albino because they have small BB. After crosses, F1 generation, they have large A, large B, small b. So, for example, dihybrid crosses, large R, small r, F1 generation, they will have large R, small r, large Y, small y. After that, we did intercrosses. So finally, we have the genotype 9 to 3 to 3 to 1. But in these cases, the large A, large B, in these cases, they will have pigmented, they will have brown color. But in these cases, small AA, large B, they will show the albino. In this case, if they have small bb, they will also albino. Albino means that they don't have any color. And small AA, small bb is also albino. So 9 to 3 plus 1 is 7. So 9 to 7 is they have the 9 is pigmented, 7 they will have albino. So this is the duplicate recessive epistasis. In this slide, we summarized in table 5.2, summarized the modified dihybrid phenotype ratio due to gene interaction. So if there was no interaction, the phenotype of, for example, 9 to 3 to 3, 1, they will follow the independent principle of independent assessment. So one encodes the seed shape and one encodes the seed color. And their final ratio will be 9 to 3 to 3, 1. So if they follow the recessive epistasis rules, This one we can see in coat color in dog. For example, if they have a large E, they will have a color. For example, large B will have the black color, and small bb will they have brown. And if they don't have the large E, they will not have any color, even though they have a large B and small bb. So in this case is 92324. How about the dominant epistasis? We can see the color in this Cauchy. So if they have large W is dominant, then epistatic dominant to large Y or small YY. In these cases, they will have the white Scotches. Also in these cases, because they don't have a large W, in these cases, if they have a large Y, they will have the yellow Scotch. They don't have a large Y, they will make the green Scotch. So the phenotype ratio is 12:3:1. How about the duplicate recessive epistasis? You can see in albinism in snail. So if they have small AA, or if they have small bb, or if they have small AA, small bb, they will have no color albino. And if they have a large B, small b, they will have a color, pigmented. The ratio will be 9:7. And also there is the interaction, but in this genetics class, we will not cover this one. We only cover recessive epistasis, dominant epistasis, and duplicate recessive epistasis. So in this slide, we will see another form of gene interaction. We call it as complementation. So complementation means that determine whether mutations are at the same locus of the same chromosome, or mutation are at different loci of the same chromosome. So this is this is difficult to understand. Now we have to, I think I will explain the complementation test. So parents that are homozygous for different mutation are crossed. For example, the father has the small AA. So, for example, small AA is the mutant form of the wild type A, and small bb is also mutant form of the wild type A plus, but they are located in the same locus of the same chromosome. So after crosses, father and mother, they crosses, their offspring will have the in the same chromosome will have the one they have A allele, and the other one they will have small b allele. So in this offspring will show the mutant phenotype. So if the mutation occur at the same locus, then the heterozygous offspring, because they have the small a, small b, heterozygous offspring have the only mutant alleles and also exhibit a mutant phenotype. So they will have the only mutant allele and will exhibit a mutant phenotype. How about in the other case, this one? So whether some mutation, small a is, in this case, small a is located in the upper region, and small b is located in the is a downstream region. Okay, in the lower region. Okay. Okay. In this case, the parents have the homozygous for small a mutant form. How about this one? This mother also has small bb. They have homozygous for different mutation. Okay, in this case in this father have small aa and mutant homozygous for mutant uh, homozygous for small aa mutation, but they have small b plus the wild type here, b wild type here. For example, mother, they have small bb mutation, but they also have a small plus A plus means that A wild type. So after crosses, this one, the offspring, will receive this one from father and this one from mother. Okay. How about in these cases, in this allele, they have small a and small a plus, they have wild type. This is the dominant than small a, so they will have the wild type. How about here? The B plus is dominant than B, so they will have the wild type phenotype. So if the mutation occurs at different loci, each of the homozygous parents possesses wild type at the other locus. And this one also has the wild type at the other locus. So the heterozygous offspring inherit mutant allele, and also they inherit wild type allele together. So they will exhibit a wild type phenotype. So this is the complementation. So complementation is determined where mutations are at the same locus or different loci of the same chromosome. Okay, determine whether mutations are at the same locus of the same chromosome or mutations are at different loci of the same chromosome. Okay, so complementation test means that parents that are homozygous for different mutation are crossed. This is the complementation test. Through the complementation test, we will know that mutation occurs at same locus or mutation occurs at different loci. Okay. In this slide, we will learn another gene interaction. For example, the coat color in dog is determined by interactions between the genes at number of loci. For example, we already learned the recessive epistasis. If they have a large E, they will have the color like black or brown. But if they have small EE, they will not have any color because they will have the yellow, because they have the small EE like this. So at that time, we only learned about the B and E loci. Now we will learn the A or S loci. So usually the coat color in dog is determined by interaction with A, B, E, S. Previously we learned about the interaction between B and E. For example, most retriever genotype is the A, agouti. What is agouti? So agouti is type of the fur coloration in which each hair displays two or multiple bands of pigmentation. For example, this hair has some multiple bands of, for example, this region is already white, this region is they are brown, and this region also white, For example, this region is white, something like that. So this is recorded as agouti. For example, retriever, they also have, this is black, the body color is black, but they have some band, like here, this is the agouti. So most retriever they have E, because E has, the dominant E has enclosed the pigmentation. Dominant E, presence of the dominant E, uh, 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 for example, dominant E allows the dark pigmentation like this. Dominant E is the enclosed dominant E is allows dark pigmentation, black or brown like this. They have the E, large E, bearing on only at E, B, and E. So they have the agouti color, and they also have SS. SS means that the spotting locus. So if they have a large SS, they will not have any spot. For example, Dalmatian, they will have SW, SW. It means that they will have the dot spotting, is the background is white, and they will have the another color, something like the brown. The dot is brown, something like that. So Dalmatian is the ASS. Some of them color is the agouti, and also they have E, large EE. It means that they will have the color black or brown. This Dalmatian has the brown color. And also they have SW. SW means that they have dot like this. The background is the white. So bearing the B and black color, so which make them those black or brown like this. And beagles, they don't have agouti, but they have the large BB. Large BB means they have the black color here, but they have some SP. They have some different spotting pattern like this. So this is we call it SP. So the important thing this one is that coat color in dogs is determined by interaction between genes at a number of loci. Previously, we only learned the B and E. Between the B and E, there is a recessive epistasis, but also in addition to the B and E, there is another factor, A and S is involved in the coat color in dog. From this slide, we will learn another subject. Sex influences the inheritance and the expression of genes in variety of ways. So first, we will learn the sex-influenced characteristics, and also we will learn sex-limited characteristics. This is also one example, and there is another example. Okay. So what is the sex-influenced characteristics? So genes that encode sex-influenced traits are inherited according to Mendel's principle, but are expressed differently in males and females. They are inherited according to Mendel's principle, but are expressed differently in male and female. For example, the presence of beard 수염 in some goat, the 염소. For example, this is the autosomal gene B plus BB, large B, small b. This is located in the autosome, not sex chromosome, autosomal chromosome. Okay, but this is dominant in the male, but this is recessive in female. So they are different. For example, in these cases, in the parent generation, the father was beardless. The genotype is B plus and B plus because they don't have the PB because they are beardless. They don't have any beard. But the women has a beard, and they are women is beard means that their genotype will be large B, small b, large B, small b, because they are recessive. Large B, small b is recessive in females. So after fertilization, so this one they will make the B plus, and this one they will make a BB. So after fertilization, so we have divided these cases in the male or female. So after fertilization, in the male case, so their genotype will be B plus BB and B plus BB. When they are the male, this one they will show the beard because they are dominant in male. But how about the genotype is exactly the same as male? But this one, in the female, they are recessive, so in female, they don't have the beard like this. Okay, so this is one case. Another case is in the F1 generation. So this is F1 generation. So in the B is they did intercrosses. So male was the B plus BB, and female is the B plus BB. So they intercrosses. So this one they will make the B plus BB gamete, and the female also make the male make the sperm. Sperm has B plus and BB, and female make the egg, which has B plus BB. So after we can do fertilization, intercrosses, so we can do Punnett care, but we have to divide the cases in the female, male and female. So in male cases, and we have divided in the female cases. So in male cases, after we did the Punnett care, 1 out of 4 will be the B plus, and 2 out of 4 will have a B plus BB, and 1 out of 4 will have the BB. How about in women? Women, 1 out of 4 will have the B plus, and 2 out of 4 will have the B plus BB, and 1 out of 4 will have the BB. So in male cases, only these cases is the B plus is beardless, they don't have the beard, but this one, B plus BB or BB, they will have a beard like this. How about in female cases? They only have the beard in the BB and BB. But this cases, in this case, they will not have the beard, but this one, they will have a beard like this. So conclusion is the 3 out of 4 of the male are beard, and 1 out of 4 of the female, they will have a beard. So this is the sex-influenced characteristics. The sex is influenced the autosomal gene, large BB, and so this is dominant in the males and recessive in females. So this is we have to think about the male cases or think about female cases. In this slide, we will see the sex-limited characteristics. So this is also encoded by autosomal gene, gene is located in autosomal chromosome, not sex chromosome, but that are expressed in only one sex. What is the difference between sex-limited characteristics and sex-influenced characteristics? For example, large BB is also expressed, both expressed in male and female, but there are some different because the large BB is dominant in the female and the large BB is recessive in female. But this one is the autosomal gene that are expressed in only one sex. They can be expressed only one sex. In these cases, they can be expressed both sexes, but they are dominant in male, in recessive in female. But in these cases, they are expressed only one sex. The very famous example is cock. Cock is sutak, and cock fiddling in chicken. So this is cock fiddling like this. Fiddle is very loud, like 화려하죠. cock fiddling is very 화려하죠 이렇게. But this is the hand fiddling in female, and also this is the hand fiddling in the male. 그러니까 여기 같은 경우에는 hand fiddling 같은 경우에는 싹이 모아져 있죠 이렇게 싹이 모아져 있죠 이렇게 팍 돌출되어 있는 게 아니고 맞죠? This is hand fiddling in the male, female, and also this is hand fiddling in the male. So recessive allele, small h, this trait is only limited to male. For example, genotype is a large H, the male phenotype is hand fiddling, female phenotype is also hand fiddling. How about the large H, small h? Because the large H is dominant than small h, the male phenotype is the hand fiddling, and female phenotype is also hand fiddling. How about small hh? Because the recessive allele small hh is only expressed in the only male, so they show the cock fiddle like this. But female phenotype is, even though they have small h, they will not expressed in the female, so their fiddling is hand fiddling like this. So this is the one example of sex-limited characteristics encoded by an autosomal gene, but that are expressed in only one sex. For example, this small recessive allele small hh is only expressed in the male, so they show the cock fiddling like this. Okay. This slide we will learn another example of sex-limited characteristics. The example is male-limited precocious puberty. In Korean, we call it as 조숙한 사춘기. So male with precocious puberty undergoes puberty at an early stage, usually before the age of 4. Usually the male experienced puberty around 12 or 13 years old. So when they have the pubic hair, and also they will have the ambic hair, they will have chest hair, and they will have hands and legs hair. But which has the male with the precocious puberty undergoes puberty. So where their age is 4, they will have the pubic hair, they will have chest hair, something like this. So dominant allele P is dominant, large P is dominant, but only expressed in the male. Okay? For example, in the parent generation, father has experienced the precocious puberty, so they have a large P, small p, And she experienced the normal poverty. The genotype is small pp. After meiosis, sperm has a large p or small p. The egg will have small pp. So after fertilization, so in sex limited characteristics, we have to divide it in the male cases and female cases. Okay, in the male cases, for example, this one crosses with this one, or this one crosses with this one, and this one crosses with this one, and this one crosses this one. So 1 out of 2 will have the large p, small p, and 1 out of 2 will have small p, small p. In these cases, the male will have the precocious puberty because the large p is dominant and small pp is normal puberty. How about in the female? Female is exactly same as this kind of genotype. 1 out of 2 is large p, small p. 1 out of 2 is small p, small p. But they will show the normal puberty because the dominant allele p, large p, is only expressed in the male. After that, okay, this is one example. Another example is here. Okay, both male and female they show the normal puberty. The father has for example, the genotype will be small pp, but mother, she has large p. She is heterozygous. But even though she has large p, so their phenotype is normal. So after meiosis, the sperm will have small p, and egg will have small p and large p. Large p and small p like this. So after fertilization, in F1 generation, so 1 out of 2 will have a large p, small p, and 1 out of 2 will have small p, small p. So in the male cases, so 1 out of 2 will show the precocious puberty, and 1 out of 2 will have the normal puberty. So, for example, in the female daughter, so their genotype is exactly same as the male. 1 out of 2 large p, small p. They will show the normal puberty. And 1 out of 2 is they will also show the small pp. They will also have normal puberty. So it means that both male and female can transmit this sex-limited trait, but it is only expressed in the male. So dominant P is only expressed in the male. So this is another example of sex-limited precocious puberty. In this slide, we will learn the cytoplasmic inheritance. For example, cytoplasmic inheritance. In Korean, we call it as 세포질 유전. For example, mitochondria, our cell has the nucleus. In nucleus, we have the chromosome. Human, we have the chromosome from 1 to chromosome 22, and at that, male has XY and female has XX. But in the cell, the mitochondria also has their own genome. We call it as mitochondrial DNA. Okay, so this is cell. This cell contains equal number of mitochondria with a wild type gene, wild type mitochondria gene, red color, and also mitochondria with mutated mitochondrial gene. It's blue color. Blue is 4, red is 4. So during the cell division, mitochondria is segregated randomly. They can be randomly segregated. So after one division, in this case is two blue is go to in this side, and 2 red is go to in this side. In the other side is also 2 blue it was here, and 2 red is here. After that, mitochondria is also can be replicated. And after that, and also nucleus is also replicated. So during cell division, so for example, the blue, 2 blue will become the 4, 1, 2, 3, 4. 2 red become the 1, 2, 3, 4. And 2 becomes 1, 2, 3, 4. And 2 becomes 1, 2, 3, 4. But during the cell division, in one cell, 3 red is located here, only 1 blue is located here. In one cell, they will have only 3 blue mitochondria here, 1 red mitochondria here. In these cases, all of blue is located in one cell, all of red is located here. So, and after that, mitochondria can be replicated. So finally, this cell has the 1, 2, 3, 4, 5, 6 red mitochondria and 2 blue mitochondria. This cell has the 2 red mitochondria, 6 blue mitochondria. In this cell, we'll have all of this cell has blue mitochondria, means that mitochondria with mutated mitochondrial gene. And this cell has all of them is the red color. So it means that this cell will have all of the mitochondria with wild type mitochondrial DNA. So usually the cytoplasmic inheritance exhibit extensive phenotypic variation because individual offspring contains various proportion of cytoplasmic gene. So they contains various proportion of cytoplasmic gene. So example is we already explained here, progeny cell that differ in their number of mitochondria with wild type mitochondrial gene or mutated mitochondrial gene. Slide, we'll also see another example of cytoplasmic inheritance. The example we can see in the red color in pork lard. In pork lard, in Korean we call it as bungot. Okay. So usually this is the pollen plant. So they have so in the answer they will have the pollen, and this is the male plant. So in this plant they removed all of the ovary, and this plant they removed all of anther. They only contains ovary like this. Okay, so they did crosses. So in the male pollen plant, the leaf shows that white, one plant shows the green branch, green leaf, and also one show that variegated. Some of them show that blue, and some of them show the green, and some of them shows the white like this. So, okay, and they have for example, also this is also from the female plant, because they removed all of the anther. They only have the ovary like this. But in this flower it comes from this branch, this gaji. The gaji they have the white lip, and also this uh, this flower is originated from these branches. They have the green color, and this flower originated from the variegated. They have mixed color like this. Okay. In these cases, they cross with this pollen and this ovary. After that, they have the white color. How about this one? They will also have the white color. How about this one? They also has white color. How about this one? They cross with the green and they cross with the white, or they cross with the green, or they cross with the variegated. They have all of them they green. So in these cases, they cross with the male plant, which has the white color, and also they cross with the green color, and they also cross with the variegated. But in each cases, some of them they showed white, some combination they show the green, and some of them they show the variegated, mixed color like this. So it means that the phenotype of the progeny is determined by the phenotype of the branch from which the seed originated. So this seed is originated from this branch. The branch's color is the white, so all of offspring is white. For example, the seed originated is from these branches. The branch's color is green, so all of offspring will show the green color. For example, the seed originated is they have this branch. They have the variegated. So in these cases, some of them they show all of them white, or some of them is green, or some of them is variegated. So it means that the phenotype of the progeny is determined by the phenotype of the branch from which the seed originates, not from the branch on which the pollen is originated, not from the on which the pollen originated. So this is another example of cytoplasmic inheritance. In this slide, we summarize the characteristics of cytoplasmic inheritance. So it present in males and females together. So usually they inherited from one parent. So in these cases, we found that typically maternal parents, but not always. Just typically, but not always. So typically on the maternal parents. And also the reciprocal crosses give different result, and also usually this one exhibit extensive phenotypic variegation even within a single family. This comes from within single family, but they show the extensive phenotypic variation. Okay. In this slide, we will see the genetic maternal effect. So in the first slide of this chapter, we learned about the shell coiling of a snail. So some of them is the left-handed, some of them is right-handed. Okay. But the maternal effect means that genotype of the maternal parents determine the phenotype of the offspring. And shell coiling, the phenotype of the shell coiling in snail is determined by the genetic maternal effect. First of all, we have to know the concept. The gene which is involved in shell coiling is determined by the autosomal gene, S+ gene. So S plus is dominant than the S. Okay. So if they have S plus, for example, S plus, their phenotype is dextral, right-handed, right-handed. So if they have small ss, their phenotype is sinistral, left-handed. Okay. Okay. Now it is the parent generation. So father was S plus, so their phenotype is the dextral, right-handed. For example, the mother is small s, and their phenotype is the left-handed. So after meiosis, they will make the sperm, in position father, they will make sperm S plus, mother, they will make the egg S. After fertilization, their genotype is S plus S. Usually the S plus is dominant than the S. Their phenotype should be dextral, but in F1 generation, genotype is S plus S, but phenotype is determined by the genotype of the maternal parent. The phenotype, even though they have S plus S, their phenotype is determined by their maternal genotype, mother's genotype. Mother's genotype is SS. That's why all of offspring will show the sinister left-handed. How about in F2 generation? So they did self-fertilization. So male has the sperm has S plus S, and egg also has S plus S. We can do the partners care, and after generation, the genotype will be the 1 out of 4 will be the S plus, and the 2 out of 4 will be the S plus S, and the 1 out of 4 will be the small ss. This is the genotype. However, the phenotype is the mother's genotype. So what is the mother's genotype? Mother's genotype is the S plus S. S plus S means that they are dextral. So all of offsprings is dextral. So the conclusion was because mother of F2 progeny has genotype S plus S, so all of F2 generation is dextral. So this is the genetic maternal effect. Genotype of the maternal parents determine the phenotype of the offspring. For example, in this case, SS was the genotype of the maternal parents, and in this case, in F2 generation, the S plus S was the maternal mother's genotype. Okay. In this slide, we will see the genomic imprinting. In Korean, we call it as 각인. So genomic imprinting means that differential expression of genetic material depending on whether it is inherited from male or inherited from female parent. The very famous example is genomic imprinting in the IGF2 gene. So IGF2 gene is located in chromosome 11. In chromosome 11, we have short arm and long arm, and IGF2 is located in the short arm. Okay. What's the role of the IGF2? IGF2 is the full name is insulin growth factor. So if the IGF2 gene is expressed, usually this gene is activate the stimulate the fetal growth. So the fetus can be growth like this. Okay, but so usually we receive the one allele from father, IGF2 gene from father, and we also receive another IGF-2 gene from mother. So usually the paternal copy of IGF-2 is actively expressed in the fetus, but maternal copy of IGF-2 is completely silent. So this is the, for example, so if there is some mutation in IGF-2, so it means that so they receive the mutation. If there is mutation in IGF-2 in paternal allele, so in these cases the fetus will die because there is some mutation in the paternal allele, so it cannot stimulate fetal growth, but mother side is completely silent. For example, another case is the paternal allele they have the wild type, there's no mutation. Of maternal allele, they have a mutation. In these cases, the fetus, it can still stimulate the fetal growth because the paternal allele is always actively expressed in the fetus, and IGF-2 is also paternal allele with wild type. But the mother side, even though there is a mutation in IGF-2, the maternal copy is completely silent. So only this one is activated, so it can stimulate the fetal growth. So this is the genomic imprinting. So differential expression of genetic material is depending on whether it is inherited from male parents or female parents. So this is usually we can see in the IGF-2 gene. So I did not explain in detail, but some background information. If you learn the epigenetics, you will completely understand this phenomenon, but I will not cover the details. So in the next semester, I will open the epigenetics class. If you are interested, if you want to learn the further detail in genomic imprinting, just you had better take a course in the next semester, epigenetics. Okay. This slide we summarize the influences of sex on heredity. Table point 5.5. So one is we learned about the sex influenced characteristics. So autosomal gene, for example, we learned in the beard, the autosomal gene, large b, small b. This one is autosomal gene that are more readily expressed in one gene. So, for example, but are expressed differentially in males and females. For example, autosomal large b, small b is dominant in the male, more readily expressed in the male, so they working as the dominant, but large b, small b, they are not expressed in the female, so they work as recessive in the female. So it means that sex influence means that autosomal genes that are more readily expressed in the one gene. Sex-limited characteristics we show in the Cox feeder. So for example, autosomal gene whose expression is limited to one sex. So small h is only limited to male. So that's why they show that co-feeding like this. And also we learned about genetic maternal effect. The nuclear genotype of the maternal parents determine the phenotypes, the offspring's phenotype. So we learned it. And we also learned the genomic imprinting. And we also learned cytoplasmic inheritance. Okay. This slide, we will learn the final subject. The expression of genotype may be influenced by the environmental effect. The very famous environmental effect was temperature. So in this slide, we will see the temperature-sensitive alleles. So allele whose product is functional only at a certain temperature. The very famous example is expression of the vestigial mutation in Drosophila. So usually the Drosophila has normal wings like this, very big wings like this. So if they have the vestigial mutation, they will have a very short wing like this, okay? But so below 29, the flies homozygous for vestigial, so they have the vestigial flies which has the homozygous for vestigial will have the reduced wing like this. But above the 29, above the 31 centigrade, around here, flies homozygous for vestigial develop normal wings like this. So because the allele whose product allele, the vestigial allele, this product is only functional is below the 29. So that's why in these cases they make the reduced wing, but above the around the 31 centigrade, because the allele from the vestigial, they will make the product, but the product is not functional at above 31 centigrade. So at that cases, they will develop the normal wings like this. So this is one example of temperature-sensitive allele. This slide we will see another temperature-sensitive allele. So another example is expression of Himalayan allele. So this is expressed in the rabbit. So rabbit homozygous for Himalayan below the 25 centigrade, they have black. But above 30 centigrade, rabbit homozygous for Himalayan don't have any pigment like this. So usually in the extremity, in the ear or nose or the foot and the tail, they have the black color because in this location their body temperature will be the below 25. So pigment in the extremity where the body temperature falls below 25 centigrade, they will show the black phenotype. Because Himalayan gene, they will encode black color. So below 25 centigrade, they will encode black color. But above 30 centigrade, rabbit homozygous for Himalayan don't have any pigment because in the center of the body, all of the temperature will be around the 37 or above the 30, so they don't have any pigment in the body, even though they have the Himalayan genotype. Okay. In this slide, we will see the concept of discontinuous characteristics and continuous characteristics. Discontinuous characteristics means that there is a relatively pure phenotype, but continuous characteristic, they have the continuous distribution of phenotype. So, okay. So one example is we can see in the Mendel's P. The Mendel's P color will be yellow or green. They have two phenotypes. So yellow or green. For example, yellow or green. And also another example of the Mendel's P. Some of them they have tall size. Some of them they have the dwarf size, short size. So the Mendel's P they have two phenotypes. One is the tall size. The other one is the dwarf, very small size. This is the discontinuous characteristics. So except for Mendel's P, all of plants, usually all of plants, So they show the continuous distribution of a phenotype. One of the plant they have the tall size, one of the phenotype they have the medium height, and one of them they will have dwarf size like this. So this is the discontinuous characteristics. So usually the gene involved in the plant height, usually at multiple loci, and multiple gene is take care of the plant height. That's why they will show this continuous distribution of a phenotype. So for example, plant height, aging is also involved in the plant height, and B gene is also plays important role in the plant height, and C gene is also important in the plant height. At that cases, they show that this continuous distribution of phenotype. In this chapter, we learned about extension and modification of basic principle. First, we learned additional factor at a single locus can affect the result of genetic process. And second, we learned the genetic interaction, gene interaction. Third, we learned sex influence. And also, finally, we learned expression of genotype may be influenced by environmental factor. Okay, so today we will finish class. Okay, thank you.